So let me welcome you again for this last lecture, which is for the course Omics Technologies. And we are trying to convey these simplified concepts for the basic biology for engineers and big data analysis. So in this last lecture for this particular course, I'm going to now give you some flavor of how different tools which we have been talking, different concepts which we have been talking, how they can be utilized for some applications. So please feel free to ask anywhere in between the questions, but also pardon us if there is a too much, going to be some part of clinical and biological type of concepts. But the idea is many technology which we talked about, many softwares which we talked about, how all of them could be well utilized for investigating these problems. So now we are trying to put it together and show you that how to make sense of those actual problem which we want to investigate by using the tool which we have been talking. So in my first part of the presentation, I will talk to you about something very relevant, which is we are all currently facing the issues of COVID-19. And my lab is also working on the COVID-19 proteomics projects in collaboration with the clinician from Kasturba Hospital and Nair Hospital and the Slok Hospital in Mumbai, where we are actually collecting patients' clinical samples and trying to investigate in different ways that what are the clues which we can identify to understand this disease better, and also probably provide some better ways of prognostic markers as well as therapeutic targets. And also towards the latter part and the final part of my presentation, I will give you what else is happening globally, which is quite relevant and interesting, which is essentially omics-based revolutions. So let me first take you to my first part, which is our actual research work in our lab, which is being done during this lockdown time, last 3-4 months time essentially. So we are going to talk to you about a multi-pronged omics investigation of COVID-19 patients to identify which are the key molecular networks of disease severity which we can identify. So again, the technologies which we are going to use are essentially proteomics and also sometimes metabolomics. So just giving you the broad overview of the COVID, I'm not going to talk anymore. I'm sure you've all heard about COVID-19 and how the SARS-CoV-2 virus works. But I just wanted to give you the feel that how these omic technologies have really contributed to understand this virus well. So very first thing which happened, as soon as people knew about that it's going to be now the big problem and pandemic, the sequence of this virus was obtained with using next generation sequencer. And from that only, now once you know the gene sequences of ATGC, the base pairs which we talked earlier, now you can develop some specific primers and you can develop the test, which is what you see now currently being used as a real-time PCR test. So polymerase chain reaction to amplify the viral RNA, and then you want to look at a specific transcripts and you want to determine if a patient is having this particular viral infection or not. So these things are possible based on the omics-based investigation, NGS-based genomics investigation to start with. Of course, then cryo-electron microscopic structures were revealed, which told about how many proteins this virus has, what is the structure of these. And then mass spectrometry-based analysis gave a lot of idea for which peptides and proteins to target. Microarray-based approaches have been used to look for which are the best epitopes, which can be utilized to develop these kind of IgG and IgM kind of assays. And of course, various ways of therapeutic assays are being used to investigate this virus and look for the possibilities of drug repurposing. And of course, then vaccine research is happening. Finally, a lot of data is being generated in the process, which is all is being utilized with the systems network type of analysis. So I'll first talk to you that we were also very keen, like any one of us right now, to see that can we detect this virus from the nasopharyngeal swab. So the technology which are currently being used are the real-time PCR. This has a lot of advantages because it is available in the clinical labs, but it also has many disadvantages. For example, the sample has to be stored in the very cooling condition, the swab sample, and then you have to amplify that RNA which is coming from the virus, and you want to then find out are there sufficient amount of viral RNA which you can detect in the real-time PCR. So we thought, can we detect the viral peptides which is present in our nasopharyngeal swab? So idea is if you take respiratory specimen samples from the patients, the same sample which is being used for real-time PCR, can we now look at different approach of mass spectrometry and proteomics and find out the peptides of virus? So that was the first interesting objective which we have. Uh, but given the way samples are collected now, the viral transport media or the VTM, where this nasopharyngeal swab is being collected, that itself has a lot of interference. So idea was first, can we optimize the protocol and find out what is the best way of these viral peptide extraction from this complex mixture. And then also, while we are doing that, can we also look at which are all human proteins present in these respiratory specimen? And also, are there some pattern emerging because of the viral infection? Are there some human protein also getting affected? And then can we develop some of the tests using these peptides and proteins? So as I mentioned that, the nasopharyngeal swab being collected from these clinicians and the clinical settings from our hospitals. Samples are collected in the VTM, viral transport media. They are doing the RNA extraction, cDNA preparation, and the real-time PCR test, which is telling patients that these are the positive or the negative of COVID. We also took the leftover VTM samples and precipitate that with different type of organic solvents like ethanol, isopropanol, and acetone, and try to precipitate the protein from this. Digested the protein using trypsin enzyme, which we talked in the workflows. Cleaned up the salt from there, and then estimated the quantification or the quantity of the peptides, analyzed them using LC-MS based approach of Orbitrap fusion or using another software like Skyline, and then looked at which are all human proteins present in that nasal sample or which are all viral peptides and proteins present. So these were our objectives to investigate. So again, there was no idea for this type of workflow. We are all working on a very new territory, a new pathogen for which there has been no literature. So we have to really start fresh. We have to really start from scratch. So we looked at various approaches of protein extraction, which can give maximum amount of viral peptide enrichment. And what we could see that, isopropanol, ethanol, acetone, these are the number of unique peptides of virus which we can detect from these methods. So we thought, why not we pool all of these possible extracts of peptide, and so that we can have good enrichment from all of these methods. So then we can have better coverage of viral peptides from these sample, and we can detect them easily. So while doing that, then we were able to look at 4 peptides from one of this protein known as spike protein, 3 peptides from the protein nucleoprotein, and 4 peptides from the replicase polyprotein 1AB. And based on these 11 peptides, we developed a test which could distinguish that if a patient is COVID negative versus COVID positive. And we also tried to correlate that with the real-time PCR values of, the high CT or the low CT value. High CT value means, the viral infection is less, and low CT value, 15, 16, or 18, indicates that there could be high amount of viral load. And those are also reflected with the peptide type of this multiple election monitoring based test. So this type of test, we are very happy that, we are able to demonstrate that this can work. So there's a lot of opportunity to look for simplified ways of extraction methodology. All we are doing, you take the swab, precipitate with ethanol or acetone or isopropanol, and then take those protein, digest them with peptides, then run in the mass spec to identify these viral peptide. If you inactivate the virus using ethanol or isopropanol, that's very safe sample to work with and very easy to handle. And that's another advantage of this method. So we have definitely overcome many of these issues, and a lot of opportunities are there. But the challenges are the instrument which we are using, the mass spectrometers, they're quite costly. They're not available in all the clinical labs because of the cost and the high end of the handling of these instruments. It requires very specialized, skilled manpower to operate these instruments. So therefore, the overall reach is limited for developing this kind of test, which could also be very cost effective. So these are the challenges, but definitely there are more options which now we are able to provide and explore, that these are possibilities of doing the viral peptide detections. Additionally, what we were also actually not at that time expected, but when we look at the respiratory specimen, the sample from the nasopharyngeal swab or otopharyngeal swab, so then we thought, are there some human protein which is also changing because of the infection from virus? So same data which we obtained from all the protein from the database, then we search for COVID negative versus COVID positive and compare those using the same software which you just saw, the MetaboAnalyst, and look at which are all proteins which are maximum differentially expressed. in these two populations. And as you can see, the top, this group of proteins are different in that the COVID negative, and the rest of these group of proteins are quite different than these patients of which are COVID positive. Also, when you are doing these kind of clinical tests and these type of assays, you have to make sure that your experiments are very reproducible, and you have various quality control checks and metrics in place. So that's where we have used a lot of control QC pools throughout the test, and those came very linear and those gave very good response. So looks like within the COVID positive and COVID negative patients, because of the virus infection itself and because of the host immune response, which is going to fight with the virus to some extent, there is a lot of protein change happening in the human proteins as well. So based on these, we are able to now identify such of these proteins, which is we have also patented, and we are also now working further to have more, the companies involved with the licensing technologies and successful translation of these type of tests. So we can see many of the clinical markers like lacto dehydrogenase and various clinically known proteins are also coming quite different in these protein-based tests which we are developing, and several new proteins have also been identified, which is quite interesting. But next thing which we wanted to really learn that are there some differences in the non-severe and severe patients? So while a patient could be negative and they can be discharged, but within the positive patients, some will be mild and many of them will actually turn to be severe. And this is the currently major problem for the clinicians in the hospital. They do not know to whom to enroll and to which patient they say, okay, go home, because the beds are limited, ICUs are limited, overall medical staff is limited, our infrastructure is limited. So if we could somehow distinguish that among the 10,000 people who are getting infected, maybe only 500 might be prone for the severe infection, then you can only isolate those groups and try to take further treatment for those in much more regulated manner. So there is a need to look at the proteins which could indicate that a patient might be in the transitioning phase of mild to the severe type of infection. So we looked at the set of protein which are there some which can distinguish these, but by looking at the nasopharyngeal swab data, we could not find very clear pattern. There was some pattern, but we could not find very clear pattern for non-severe versus severe group of the patients. But it definitely indicated overall that in COVID-19 patients, many proteins are quite different in the expression values in COVID positive versus COVID negative. And those belong to some of these pathways like neutrophil degranulation, translation mechanism, protein metabolism, some of the platelet activation related proteins, and various response to cellular stress proteins. So many of these proteins are where most of these changes are happening in COVID positive patients versus COVID negative patients. So looking at the data from the nasopharyngeal swab, there is definitely some opportunity to look for COVID negative and COVID positive and to monitor the disease severity. But of course, the tests which we can develop on this are based on the mass spectrometers, which will have the similar limitations of the cost and the specialized skilled manpower. And also handling nasopharyngeal swab for doing this kind of test is going to be challenging, given that you cannot monitor the nasopharyngeal swab after 3 days, 5 days, and very routinely. You don't want to take that sample. Blood is being taken very routinely for all kind of patient monitoring, but nasopharyngeal swab is only taken once to initially for testing the viral infection, yes or no answer. So we also, in the meantime, we were looking at large number of patients, 100 plus patients, for which we also got the plasma samples of these COVID-19 patients. And we have been doing a lot of work in the plasma proteomics. And recently we published this paper in Communication Biology of Nature, where we showed in malaria patients what are the possibilities which can lead to the severe type of malaria. So we already had good understanding and know-how of working on these kind of plasma samples and investigate infectious disease using proteomics type of technologies. So now we were quite motivated to investigate the plasma sample of COVID patients, and overall study size was almost 180 or so patients. But for this particular proteomics study, the numbers are mentioned here. So we have now COVID-19 non-severe and severe patients, which are all positive, and then we have some COVID negative patients. Then we took their plasma sample, removed the highly abundant proteins, which is like albumin and IgGs, deplete those, did the digestion with the enzyme trypsin to make peptides, ran on this instrument of orbitrap fusion, generated all the peptide spectra, used that for database search, Then software like Analyst, which you just saw, was used for the data analysis. The peptide which we selected were used for further developing the test, which is multiple action monitoring based validation test. Some of the identified proteins and pathways were also used for testing with the possible repurposed drugs to show that which could also be effective. So to give some more insight of this work, when we look at now the plasma sample of COVID positive and negative, now as you can see, these are very distinct signature we could see from negative versus positive when we are looking at the plasma sample, and these are forming good cohorts in the PCA. And we could see a very interesting protein which is quite different in these two group of patients, and it is showing the now the subset of the protein which looks much more influential of distinguishing these group of patients. So based on these findings, then we thought, can we take some of these and do some sort of clinical translation which could be utilized by the clinician and that can be further used. So we looked at where these proteins in our different pathways and biological system, where they belong to. And then based on those, we rank those and we try to see like where they are fitting well. And some of these proteins which are shown in the insets in these box plots or the violin plots are also some of the known protein markers which are currently being used in the clinics. But we can see now much better and bigger and clear picture of what is happening. There are different type of pathways where most of these proteins are changing in these COVID non-severe versus severe patients, and can we use that to develop these tests? So now we are developing these tests, which is known as the SRM-based test, selective reaction monitoring or multiple reaction monitoring based test, where now you can see like these proteins and their peptide in non-severe and severe patients, how they are looking, very different intensities one could see for multiple proteins. And similar kind of analysis is being currently, being done on the large number of samples to see can we make it more clinically applicable test. Then we also looked at which of these host protein and virus proteins have some interactions using some databases. And further, we were also keen that now we know that which of the major proteins are changing in the host. And there are a lot of FDA approved drugs for which can be tried to target and see can they inhibit these proteins and are they able to dog these. So then we looked at some of our interesting protein like serpin D1 and serpin A7 and S100A9. We use this drug, selinexor, which is FDA approved drug, and which were able to bind very well. So what we have just showed is only those protein and drug pairs which are able to show very strong binding. Others we tried for many others which did not show binding, so those we have not presented here. But it looks like that if a cocktail of some of these drugs or these drugs alone in combination with other drugs which are being given right now could be tried out by the clinicians so that they could try to control the disease severity at the early stage. But of course, this part of the work is very preliminary because for doing any kind of drug to the patients, you need various level of trials. And good thing is that these drugs are already FDA approved, so it means they have been tested for the toxicity and side effects of the patients. But still, in this context, it will further require a lot of testing before it can be clinically approved for taking it forward. But this type of data from the big proteomics and omics data not only gives us the glimpse of what is happening and changes in the patient, but also could develop the tests which could indicate these type of progression and also provide us the targets which could be utilized for potentially giving some therapeutic aspect as well. So in addition to these things which I just showed you the flavor of the work, we are also doing various other type of analysis from the proteomics side, metabolomics of these patients. We are also looking at FTIR based investigation, microarray based tests, so variety of things which we are doing to investigate this virus and understand it much better. So from this part of the presentation, I hope I was able to convey you that different technology which we have been talking can be very useful, even something like in the case of COVID, when we did not have any idea, we are able to detect these type of virus from the patient samples. And also we are able to monitor the human protein response to the disease severity, which could indicate a patient might be moving from the mild to the severe group of the severity. We have filed these two patents already for this work, and two manuscripts are already in the process. One is under revision, another is already submitted a couple of days ago. So, but there is a lot of work which we are doing, which is not keeping in mind patent or publication, but rather can it be clinically employable and clinicians could benefit, patients could benefit from this work. So, in this phase of COVID-19 pandemic, I'm sure all of us had seen a very, big transformation, lot of changes happening around. But at the same time, I'm sure, science education has really taken a different turn with more virtualization, more e-learning, and even the course which we are doing right now for TQIP is another indicator that despite, of these challenges, you can do a lot of learning by sitting at your home or in sitting in your offices, and we can still connect virtually, and we can do a lot of collaboration and a lot of learning, which is a very new way of thought process, which was available earlier but was not utilized so well. So in the pandemic, we have also learned many positive things from this phase. So this pretty much ends my first part of the presentation. I will now spend next uh, 15-20 minutes time on discussing about what else is happening overall in the world, which is linked to the omics technologies, and which I think, as a part of this course, it is a good idea for you to know that, what are the key opinion leaders they're working and which type of technologies and which are really transformative in nature currently being used in the world. So first of all, the area which we work on is proteomics, where the organization is Human Proteome Organization. To get more detail of that, you can go to this website, hupo.org. This organization is kind of providing an umbrella to take various initiatives for Human Proteome project, and there are different teams involved for the chromosome-centric Human Proteome project and the biology disease-driven Human Proteome project. And there are different clusters, which is, different scientists are working in different areas, from mass spectrometry technology to the knowledge cluster to using antibodies or looking at pathology pillars. And then, as I mentioned, there are different chromosomes. For every chromosome, there is a different country who is involved to look at all the proteins belonging to those chromosomes. And there are different teams who are working on biology and disease-driven human proteome projects. So these are different ways of looking at the bigger picture of the human physiology and human proteome. And we just recently published a paper on the high stringency blueprint of the human proteome from the HUPO, which is 90.5% representation of the human proteome. Another important portal here is HPA, or the Human Protein Atlas. So if you go here, you can search for a given protein which is of your interest. You will find a lot of interesting information. They are publishing really high quality data in science every year. This is the tissue atlas, cell atlas, pathology atlas, brain atlas, blood atlas, metabolic atlas. So you go there and you will find, almost all the proteins given for that particular objective, and you can get lot and lot of information from here. So for example, this group has published this tissue-based map of the human proteome, Dr. Matthias Ullén and his team from Sweden in 2015 in Science, where they showed that which are all proteins in specific organs are present using this kind of antibody-based approach. And more recently, they have published all the pathology atlas for the human cancer, which represent almost 17 different type of cancer and which of the transcripts which can be located in these different organ and their cancer. So all of this data could be very well obtained and analyzed and utilized by going through HPA portal. Other major initiative happening globally is Proteogenomics, which is how to integrate data from the genomics and proteomics technologies together. Because idea is that, as if you go back to the previous discussion about central dogma of life, information has to move from different levels, from DNA, RNA to protein. Many things are part of the blueprint, lot of mutation and changes happening at the genome level, and many things only appear at more at the functional level, at the protein level. So if we can integrate that data, that information under proteogenomics, that could be very powerful. And this is more powerful, especially in the context of cancer, which is having a lot of mutations, and but those are not sufficient to do all kind of treatment. So what else one could analyze to get more layer of information? That's where proteogenomic tools are becoming more and more handy. And it starts from, as I said, DNA, RNA, and protein investigation. At DNA analysis, you can look for whole genome sequencing or whole exome sequencing. For RNA, it is RNA sequencing, and protein, you can use LC-MS/MS based approach. So you are going to generate data at genome level, at the transcript level, and at the protein level, and trying to integrate those together. I am not going in detail about how to do that process, but there are a lot of details coming from the National Cancer Institute of NCI, where series of good papers in Nature and Science are getting published on proteogenomic characterization of different type of cancers. This is just one example of breast cancer. So breast cancer, as I referred earlier as well, it was thought it is one cancer, but it is not. There are 4 different subtypes, basal-like, HER2, luminal A, luminal B. And now if you want to further, identify what is the gene signatures of these, what is the protein signature of these. So scientists are now doing, integration of proteogenomics. So where all of these, samples from different women who are suffering from breast cancer are taken, and every sample is analyzed to look for the mutation, copy number analysis, RNA sequences, MSMS level protein analysis, MSMS level phosphoprotein analysis, and reverse phase protein array based analysis. So all of these different technologies are being used from the same sample to generate different type of data, as you can see now. So what it actually turns out, now you can see the different patterns emerging for different groups, which is what indicates that they're all different. Like this group is quite different for these genes versus this group is different for this gene. And this is the type of information which you want to generate to really do much better treatment for these patients, which are otherwise really getting affected if you are not able to provide very unique clues for that. So Joe Biden now, he does not need any introduction. He is currently the president of the United States. At the time of Barack Obama's presidency, he was vice president, and that time he actually initiated Cancer Moonshot project in the US. His goal was, can we increase the the pace of the cancer research so that the patients who are really affected could at least benefit much faster. If not in 20 years, can it be done in 5 years? Can you accelerate the pace of that? And based on his initiative, of which involves pretty much the proteogenomics integration, next thing was to make an international consortium, which is ICPC, International Consortium for Proteogenomics Analysis. And so this consortium looks for various international different groups of countries involved. And we are very proud to be part of this group from India now, which India became 12th country from IIT Bombay and Tata Memorial to be part of this Cancer Moonshot and ICPC project. Other initiative which is happening globally is IPOP, or Integrative Personalized Omics Profiling. This project is being done at Stanford University with Dr. Mike Schneider as the director over there. Idea is, a healthy individual, you take their samples at different time interval. It's like you go to a medical hospital and your blood sample, urine samples are collected, and that gives you some very basic parameter for the biochemical test, which indicates that at least patient, an individual is behaving normally, or is there anything happening, any problems are happening. But now in this case, data is looked at a different scale. Rather than looking at only the the basic biochemical parameters, can we look at entire omics profile? Can we look at all the metabolites, all the proteins, which is changing in these individuals over the time period? So Dr. Mike Schneider, he did the experiment on himself for over 14 months time period, and his samples were collected and same samples were analyzed on the longitudinally. And what really it indicated was something very different, that he's quite thin person and did not have any indication of diabetes, but his protein profile and metabolic profile pathways indicated that he is getting prone for diabetes. And actually after that, or after 2 months or so, he was actually confirmed to be diabetes from the clinicians. But omics data indicated much ahead of time than actual clinical test could actually indicate. So something like this could be good way of doing the even wellness profile of individual. Why only go to doctors and do the test when you are actually sick? Why not to look at the wellness index of an individual who are healthy? And this is something which is work being done at ISB, Institute for Systems Biology in Seattle, where they just published this work in Nature Biotechnology. This work is done in collaboration with the company Arivale, which is a spin out from there. And they took 108 healthy individuals, and looked at their samples longitudinally, as you can see, over a 9-month time period. These samples being analyzed and in very much consultation with the coaches and the medical nurses and the doctors, as well as the omics scientists. The entire data really gave a lot of indication that some people were having cardiovascular diseases, some were having diabetic risk, and how they're behaving and how they're going to get more prone for any of those problems. So now you can think about where the entire field of medicine is progressing, where the entire field of all the clinical tests are going to go, given that now there is more access to these omics technologies and big data analysis. I think our test, which we used to do for 5-10 biochemical parameters, is changing now to the thousands of features of omics data set, from the genome to transcriptome to the proteome. So all of this is generating a lot of big data, as I'm sure you're aware of, that the technologies are very robust now. One could use them easily, and they are quite accessible to different centers in different parts of the world. So one could generate a lot of data along with various imaging data, which is also being generated, like radiology data, immunohistochemistry data. All of that is generating a lot of big data, which is not easy to handle, and it's actually opening up, on one hand, a lot of opportunity and a lot of challenges as well. But where we are progressing in case of medicine, in the future aim is to look at every individual as a unique individual and look for more personalized treatment of the patient. So if you can look at breast cancer not as a one tumor type, one cancer type, but look at which subtype it belongs to, and within that subtype what that one individual may have variations and may have a specific mutation, may have some modification, can we try to capture that and give the patient a specific and more precision medicine-based treatment based on the person's genetic and protein profile. And that is really going to be very targeted, very much applicable to the individual, and people are going to see very different type of positive response based on these future precision medicine. So data size and our computational powers are many times are quite limitation of doing this kind of entire work. This part of the work is still getting more progress. That has not reached very easily to clinics as of now, given the instrumentation, which is quite costly. This is next generation sequencer. This is one of the mass spectrometer, and also the the specialization required people to handle these type of technologies and the big data which is being generated, how to handle that, analyze that. All of these are the challenges, but results are very promising, which is makes it really going to be futuristic approach of taking these technologies forward for the precision medicine. Uh, but given all the facts which I spoke to you, question is, we are able to generate a lot of big data from IIT Bombay, and likewise many labs from India and abroad are generating good big data. How are we going to use that big data and use it for societal development? And I must say that our thought of doing these kind of courses and workshops are also to link people so that they can start appreciating the what they can easily access this kind of data, and they can start using them for different type of questions, using their own type of problems of interest which is they want to work on, and also utilize their own skills of doing that analysis. So we should definitely be thinking about making that community where we can start doing data sharing, look at the proper data analysis guidelines. Data can be properly stored, but also being shared. And of course, as I said, all this is not easy, because what we are talking about in this case, human sample, which is having a lot of ethical issues, and you can't just share human information easily. So the data has to be really restricted in that way and has to be anonymized so that you cannot reveal the identity of the patient and individual. Many of these issues are there, but I'm sure you are excited that there are a lot of possibilities which is opening up in this space. So I hope with this lecture I'm able to convey you that omic technologies have really made a huge significant contribution for the functional biology and the translational research. But of course, we need more interdisciplinary approaches. We need more collaborations so that we could have significant contributions for the humanity based on these type of technology, which has so much potential. So finally, let us make a strong community, and some of our efforts from IIT Bombay has been in this direction. As I mentioned, we are part of this project, which is ICPC, International Cancer Proteogenome Consortium, where different countries, almost now 13 countries, India became 12th country to be partner of this, where from India's side, we are going to look for breast, cervical and oral cancer and look at their genome and proteome signatures to compare those profile, and then also share the data with other 12 countries and investigate and discuss those findings. We are also having a center when we are working with Institute for Biology in Seattle team to look at more clinical translation MS-based assays. Of course, we find the Skyline usage made by Brendan McLean from the University of Washington quite user-friendly, and we are many of the MRM assays based on this software. We are also very much of opinion to share our knowledge what we have and the technologies and resources what we have more in form of the e-learning and the virtual lab, which has been also highlighted in Nature and other journals based on our curriculum which we have developed here. And finally, as I mentioned to you that it's not only us, but I think all of us have to think about how to access this kind of omics data and make them more accessible for the overall community and make best use of this data. Data generation is not easy, as you can understand. Collecting the patient's clinical sample and going through the entire process of those protein extraction and the processing of the samples, running them in these complex instruments, generating data till you reach data generation, that itself is required humongous amount of work. After that, once you have generated this kind of high quality data, that should not be just kept in the isolation somewhere. And that's where the entire community is thinking more and more that how to make the data more accessible, more open, data sharing policies. So all the journals, when you are now going to publish in big journals, they are asking you to upload your data first to make it available to the reviewers. And after your data has been reviewed and you are publishing your paper, it is now being made publicly available so that anybody can access that data. So if you are aware of the tools which we have been talking in the course for different type of docking study, different interaction analysis, sequence alignments, looking at the MS secondary data analysis using metabonalyst, many of those could be very well utilized from your institutes, from your students, just by sitting in your classroom. You don't have to go through generating data because data is already available. We can provide you the resource and links from where you can download the data. It's just now how best we can bring engineering skill and different mathematical skill and different analytical skill to make best sense of the data and think about big data as the more image processing type of problems and big data problem. And many times when we are working with the professors from mathematics and computer science at IT Bombay, a lot of students are doing the projects on these type of data where for them it's just the data which is in the Excel sheet, and you have been given them different cohorts A, B, C, and they have to make some sense of that and using independent approach, and that becomes a big data problem for them. But finally for us it's very important because we have to think about is that data making any sense which can also correlate to the clinical data set, and that's where collaboration comes very much handy for this activity. We then talk to our clinicians and the doctors who are partner to these projects, and large number of doctors actually working with us. Different MD students are working on these projects with us, who are then invited and discussed to review the data, and then we try to take the data forward. Let me close this session with showing you this clip, which is Joe Biden's speech, which he gave in 2017 in Human Proteome Organization Conference, where I was invited. We had close to 100 people who were invited for this gala dinner evening, and he gave this very nice lecture, but I'm just showing you 1 minute clip of that.
Not to hope on this side of the grave, but then once in a lifetime, a tidal wave of justice rises up, and hope and history rhyme. And I can't be with any more prominent group of people who may be responsible for making hope and history rhyme than all of you in this room today.
I'm not sure it was very clear audio, but he was referring to all the people sitting in that room who are all proteomic scientists or proteogenomics scientists, and trying to address them that such big and complex problem like cancer, if you are not able to contribute, who else will do that? So you have to really step up and do try to contribute so that whole humanity can benefit. So it was really touching and very appealing, speech what he gave, and that I'm sure also is going to really accelerate the cancer research and proteogenomics research overall in the world, and effectively in US under his leadership. But that similar mindsets are required in the whole world to really adapt to these new technologies. Also, I think all of you who are from engineering faculty members from different institutes have come forward to take this course. Also shows that we have to break the boundaries of clinicians and biologists and the technologists and engineers. We have to work on the big problems with very, very new outlook, with the newer approaches, without any type of bias in our mind. And of course, there's no boundaries. We have to break those boundaries and create these opportunities together. Then only I think we can solve many of the complex problems which are all lying and standing in front of us. COVID is just one of those. tuberculosis and cancer and many other major problems of healthcare sector, and of course likewise many other problems are there which could be solved at least to some extent if we all work together and bring our interdisciplinary strengths and different minds and different specializations to work on these problems. Thank you very much for staying for this course, attending this course. I am sure and we are excited that you are not only learning these things yourself, but you're also going to inculcate this knowledge and take it forward to your own students, your own classes, your own institutes, which is going to generate more ideas and more scientists who can actually think about that interdisciplinary approach of utilizing the different technologies and different biological problems and work on those together without any hesitation. So thank you so much for attending this course, and I'll be now open for any questions and discussions.